Hello, I am Vidya Patil from Yashwantrao Chavan Institute of Science, Satara. Today I will discuss about the heme group. Many porphyrin containing metalloproteins have heme as their prosthetic group. These are known as heme protein, or just slight difference in the spelling as a HAEM, heme protein according to the British English, and again some difference in the writing as a hemoprotein, or separate word as a heme protein or heme protein with slight difference in spelling. So any word is correct one. They are very large class of metalloproteins. Important types of the heme groups as heme A, heme B, heme C and heme O. In myoglobin and hemoglobin, heme B is found, which is shown in the diagram as a myoglobin cleft contains, at this position myoglobin cleft contains heme, while in this diagram there is a 4 subunits of hemoglobin. There is also 4 heme groups are present in the cleft. Just see in this diagram here. Okay, then the structure of heme is typically ferroprotoporphyrin ring. The heme consists of organic part and inorganic part as a iron. The organic part contains 4 pyrrole groups which are joined by the methyl bridges and formation of tetrapyrrole. Then 4 methyl and 2 vinyl, 2 propionate. These total 8 groups are attached to the tetrapyrrole ring, which is shown in this structure. Okay, in this structure there is a 4 methyl groups, 1, 2, 3 and 4th one. These 4 methyl groups are attached, while 2 vinyl, this one 1, 2, and these 2 propionate groups are present in the total structure. So it is a protoporphyrin type of isomer, and in biological systems protoporphyrin 9 is present. Iron atom is present at the center, which joins to the 4 nitrogen atoms, and at this position, see just here, 4 nitrogen atoms are attached to the heme, while there is a 5th and 6th coordination position. At 5th coordination, proximal histidine is bound, while at the 6th coordination there is a distal histidine is present. It is not directly bonded. This coordination is near to the distal histidine. Then there is a separate video for how to draw the heme structure. Okay, here 2 polar histidine molecules of heme chain, means there is a highly polar propionate groups of heme group are present on the surface of the molecule. At physiological pH, these propionate groups are charged groups, means CH2, CH2, COO minus. Otherwise rest of the molecule is present in the cleft of the protein. Just see here, what are the iron atom, it is bind with the 4 nitrogen molecules, while its 2 bonds, 5th coordination binds with the proximal histidine molecule, and 6th coordination is near to the distal histidine molecule. Then there is a iron atom. It can be in the ferrous, means +2, or ferric, means +3 in state. Only ferromyoglobin, means iron atom with +2 oxidation state, it can bind with the oxygen. That means for the function it is essential. And the heme group is located in a cleft of the myoglobin and hemoglobin. This proper binding in a cleft is essential. As I told, two propionate molecules are present towards the surface. At the fifth coordination, histidine is present, proximal histidine. Sixth coordination is present near to the distal histidine. See, in this diagram there is a myoglobin and hemoglobin. Myoglobin contains heme group, hemoglobin contains heme group in each subunit. Physiologically admissible forms of the heme have been investigated. Therefore, deoxymyoglobin, oxymyoglobin and ferrimyoglobin conformations are observed. They are only differ with respect to the sixth coordination. At the sixth coordination position, in ferrimyoglobin there is water molecule is present. In deoxymyoglobin it is empty, and in oxymyoglobin there is an oxygen atom is present. For hemoglobin there are also physiological forms as a oxygenated hemoglobin and deoxygenated hemoglobin. Just see, physiological forms of the hemoglobins are also investigated as a deoxyhemoglobin and oxyhemoglobin. In case of deoxyhemoglobin, just see this diagram. In case of deoxyhemoglobin, sixth coordination is empty. There is no binding of the oxygen. While in oxyhemoglobin, see this diagram, at the sixth coordination position there is oxygen is present, and this is the distal histidine. Okay, already proximal histidine is present at this side, plane side of the ring. And in this way there is a deoxyhemoglobin and oxyhemoglobin formation is possible when heme group is present in hemoglobin. So this molecule is very, very important for the function of protein as a prosthetic group. Okay, how to draw the heme group? For this there is a separate video is present. Now after this discussion I stop here and thank you.